Hey everyone, and welcome. Today, we are diving into a really cool genetic mystery. And it's one that starts right in your kitchen, but ends deep inside our own DNA. It's a story all about numbers, and how changing just one little thing can mean the difference between, say, a perfect seedless watermelon and, well, a human life.
So, think about this for a second. You slice into a big, juicy watermelon. You're all ready for that refreshing summer treat, and you notice something's missing. No seeds. How does that even happen? Is it some kind of magic? Nope, it's genetics.
And it's not just watermelons, right? Think about the bananas you grab at the store, or those super convenient seedless grapes. This awesome feature isn't an accident. It's actually the result of us intentionally tinkering with a plant's genetic blueprint. And the secret? It all lies in their chromosomes.
Okay, so to really solve this mystery, we first need to get our heads around a really powerful biological concept that's at the heart of it all. The idea is called polyploidy.
So, what on earth is polyploidy? Well, most animals, including you and me, we're what's called diploid. That just means we have two complete sets of chromosomes. We get one full set from each parent. Polyploidy is when an organism has more than two of those complete sets. We're talking 3 sets, 4, 6, maybe even 8 or more. And the word to remember here, the really crucial word, is sets.
Let's think of it like a deck of cards. A diploid organism, like us, has 2 perfectly matched decks. Super easy to pair them up and deal them out. But a triploid organism, that guy has 3 decks of cards. Now you try pairing all those up, it's going to get messy, right? And it turns out that mess is exactly what we're looking for.
The specific type of polyploidy that gives us our seedless fruit is called autopolyploidy. And all this means is that the extra sets of chromosomes all come from the very same species. It's kind of like taking a plant and just convincing it to make extra copies of its own genetic instruction manual.
So, how does this actually work? Let's use the banana as our guinea pig here. A commercial banana is triploid. It's got 3 sets of chromosomes. So when that plant tries to make seeds, which is a process called meiosis, those 3 sets try to pair up. But you can't neatly divide 3 into 2, right? The pairing just fails, the chromosomes can't separate the right way, and boom, the plant can't make viable seeds. Voila! You've got a seedless fruit. And what's so wild is that this reproductive error is a massive win for agriculture. See, plants with these extra chromosome sets often pour all their energy into just growing bigger instead of making seeds, and that can lead to larger cells, which means bigger fruits and just more robust plants overall. You see this all over the grocery store, from those giant strawberries with 8 chromosome sets to the potatoes and sugarcane that literally feed millions of people.
So that's what happens when a species just duplicates its own chromosomes. But what if you start mixing sets from different species? Well, that's where things get even more interesting, because that can lead to the creation of something completely new. This whole process is called allopolyploidy. It kicks off when 2 different, but usually kind of related, species cross-pollinate. The offspring you get is a hybrid, and it's almost always sterile. But in the plant world, something really rare can happen. The entire combined set of chromosomes can just spontaneously double, and that instantly creates a brand new, totally fertile species. This isn't just some theory, you guys. It's the epic origin story of wheat.
The story of modern wheat is just incredible. It started thousands of years ago when 2 wild grasses crossed and made a sterile hybrid. But then, this random genetic fluke happened. Its chromosomes doubled, and that created a new, fertile species. But the story doesn't even end there. This new plant then crossed with another wild grass, and you're not going to believe it. The doubling happened again. The end result of this amazing journey is the modern bread wheat we have today, an allohexaploid with 6 sets of chromosomes, carrying the genetic history of 3 different ancient ancestors.
Now you might be sitting there wondering, does this ever happen in animals? The short answer is, not really. When different animal species cross, like a horse and a donkey, you get a sterile offspring, in that case, a mule. That critical step, the chromosome doubling that makes plants fertile again, it's just not tolerated in animals. And this is a really fundamental difference that's going to set the stage for our final chapter here.
So let's pivot away from the plant kingdom and bring this story home to us. What happens when chromosome numbers change not in wheat or bananas, but in humans?
This is the core question, isn't it? If adding entire sets of chromosomes makes plants bigger and stronger, why is a change in chromosome number in humans so often linked with serious health conditions? The answer all comes down to the scale of the error.
See, in humans, the problem is usually not polyploidy, having whole extra sets.
It's a different condition called aneuploidy, and this is where a person is either missing or has an extra single chromosome. So instead of getting a whole extra deck of cards, it's like you just get one extra ace, or maybe you're missing a king. And that seemingly small change creates a huge imbalance.
And that right there is the fundamental difference. Polyploidy is all about balanced whole sets. Aneuploidy is an imbalanced addition or subtraction of just one single chromosome. One process creates superplants, the other is the underlying reason for many genetic disorders in people.
So where does this imbalance come from? Well, it's basically a mechanical error that happens when sperm or egg cells are being formed. It's got a technical name, nondisjunction, which is just a fancy way of saying the chromosomes didn't separate like they were supposed to. One cell ends up with an extra copy, and another one ends up with none at all.
And the consequences of this single chromosome error are pretty significant. An extra copy of chromosome 21, for instance, results in trisomy 21, which you probably know as Down syndrome. Other examples include conditions like Turner syndrome or Klinefelter syndrome, which involve the sex chromosomes. Every one of them is the result of that subtle but incredibly powerful error of one. Okay, so as we start to wrap this up, let's bring it all back together. This entire story, from seedless fruit to human health, it all hinges on one simple but really profound distinction: the difference between adding whole sets and adding single chromosomes. So let's just break it down one last time. Polyploidy, that's adding entire balanced sets of chromosomes. It's a process that plants have totally harnessed to become bigger, seedless, or even to evolve into new species. In contrast, aneuploidy is the gain or loss of just one single unbalanced chromosome, which in humans throws development off track and can lead to genetic disorders.
Ultimately, it all comes down to this. One is an error of whole sets, and the other is an error of single chromosomes. That one distinction, it changes everything. It's the reason why one is a powerful tool for agricultural innovation, while the other is a really important matter of human health. So, from the bananas in our kitchen to the very blueprint of our own bodies, the number of chromosomes an organism has tells this rich, complex story of evolution, farming, and health. And it really makes you wonder, right? What other profound genetic secrets are still out there, just waiting for us to discover them? Thanks for tuning in.